Shall I start, Prathamesh?
Yes.
Okay. Good afternoon to all. Prathamesh, can I start? Yeah, thank you. Good afternoon to all. A student is not a container you have to fill, but a torch you have to lighten up. And here we are with the next session. And I am Megha Hundekar, a research scholar from Mysore University, and we are here to start an informative session today on all about APT testing and research company. It's my privilege to welcome and introduce our speaker, Dr. Kishori Apte ma'am. I welcome you, ma'am, from the entire MBSI Society. Dr. Kishori Apte ma'am has completed her post-graduation and PhD from Pune University. Her professional details include many layers of experience. To highlight a few, I would like to introduce a few amongst them. She has been a proprietor of National Toxicology Laboratory since 1993 to 2014. Ma'am also served as a research associate at Indian Institute of Toxicology, and at present she is the director of APT Testing and Research Private Limited from 2015 till date. Ma'am is also awarded with Ramabai Joshi Women Entrepreneur Award for the year 2014. She is also a member of Society of Toxicology USA, and ma'am is also an IRCA certified lead auditor. Ma'am has worked on many projects, and she also has 34 national and international publications. Last but not the least, ma'am has attended innumerable conferences and workshops. It's a pleasure to have you for today's session, ma'am. I welcome you again for the session, and I'm happy to welcome the attendees for today's session. I request Dr. Kishori Apte ma'am to take over the session. Ma'am, you can take over the session.
Yeah. Good afternoon to all. Thank you for the opportunity to give that you have given me, and whatever you have said about me, I don't think it is. It should be said in that same manner. It is like we are doing work, and everybody has his or her own way of working. So we all are together on the same page, I would say. So now I'm sharing my presentation, please. So now first,
Kishori ma'am, you have muted yourself. Please unmute yourself, ma'am.
Unmute. Yeah,
Unmuted.
Can you hear? Can you hear now?
Yes, ma'am, you're audible.
Yeah, I'm audible. Thank you. Now, firstly, I would like to introduce APT Testing and Research Private Limited. This is a company which I founded in 2015. I have been working, like you said, my experience is I've started National Toxicology Center in 1993, and I'm a one person army. So I wanted to have somebody with me, like suppose something happens, somebody will take over my company, proprietor company. So I formed it into a private limited company. Now in the private limited company, I'm doing regulatory toxicology and some R&D for companies, because my trust cannot do work for as commercial. So that was the basic of National Toxicology Center, because I worked with Dr. Bhide's Indian Institute of Toxicology for 10 years, and that is what I could do. When I moved out from there after getting my PhD, what was in my mind? What can I do? I can do only toxicology, and I've done my pharmacology. So as pharmacology and toxicology go hand in hand, it becomes very easy for me to work in the same field. So I started NTC and then converted that into APT Testing and Research Private Limited, where we are doing, like I said, regulatory toxicology. So it has all the guidelines are set. We have to do the experiments as per guidelines. So there's nothing we can divert other than some research activity for companies. And while I was working with Dr. Bhide, I did my PhD. So I thought when I made my own company, National Toxicology Center, I said I should give some preference to the people who are working with me, the staff who is working with me. So I started APT Research Foundation in 2006, so that they have the facility to go get higher education, that is the either what do you call that, post graduation and PhD. So I will now talk about APT Research Foundation. APT Research Foundation was founded in 2006. It is like for the students, of the students, by the students, like that I would say dedicated to the students. And I've taken, as I have, being a pharmacologist and a toxicologist, I have an animal house facility in vivo and in vitro also. Now, in addition, in vitro was added. So when we have normal animals and animal house, I can have an array of diseases which I can induce and conduct the work on these animals. CPCSA was founded in 1999. I had already started in 93. So I know the initiation of CPCSA now, which is called CCSA. And we have all the regulatory certifications. So there's no way that I cannot conduct the studies on animals. So that is my core. In vivo testing is my core. And I think I would, sometimes I would maybe not do regulatory toxicology, but I would definitely do research because that is very interesting and we can go ahead in science by doing different kinds of research. So now coming to APT Research Foundation, the address is mentioned here. And it was established in 2006. I have got recognition as a PhD center from MUHS Nasik, that is Maharashtra University of Health Sciences, and Symbiosis International University, Pune. We had the recognition from Pune University, but then there was they wanted me to have guides for that department, different departments, which I was not interested in getting that information. I wanted my center to be recognized as a research center, but they did not give me the recognition. So I don't have any recognition from University of Pune, though all the departments from University of Pune come and do their work with me, their projects with me. Microbiology, which is that other one? University of Department of Health Science,
biotechnology,
biotechnology, IBB. Most of them do come and do their work here. Zoology, botany, they also come. But I have not got recognition as a PhD center from University of Pune, hoping to get that in near future. Now, what is the vision? Now, like nowadays, this is all mandatory, I would say, that you have to have some vision, you have to have a mission. So as we came into the corporate style of working, this was what we had. So we visualize ourselves achieving research solutions along with scientific advancement in the area of pharmacology and biological research. That is our vision. So if there are some models required by the students or the PhD students, so I'm more interested in developing the model, developing new areas of research, and that is how I'm going to, what would you say, increase my span of research and also help the students. And the mission is to offer high quality services in the field of toxicology and pharmacology. Now, when we do research in my center, in my institute, we see the efficacy and safety, because being a toxicologist, safety is also very important. Only efficacy has got importance, but even the safety later is more important. So any student who comes to my center, my research center for project, we do efficacy and safety, because without that we cannot go ahead. The new product or the new molecule or the new what would you call that, the new extract that is to be used for studying, that has to have safety so that it can be taken ahead for use in human beings or larger animals. So that is the importance of toxicology in research. Now, this is a certification that I've got, DSIR approval. And what are the kinds? Now, when we come to toxicology, different kinds of toxicity tests are conducted: acute, long-term, chronic carcinogenicity, where reproductive toxicity is done, developmental toxicology, dermal, ocular, neurotoxicity, and cytotoxicity. These are all the different kinds of toxicity studies which are as per regulations. And we can do this for agricultural products, aquatic chemicals which could be used, which would come into contact with water, soil. So we call that as ecotoxicology. Industrial chemicals that are used and then sent into the effluent, they are tested. Medical devices are tested. occupational and regulatory toxicology is also usually done. So these are different avenues. So depending on the product, what would be the end use in human beings, the studies can be selected and conducted. Now, what is the animals available here? I have mice, rats, rabbits, guinea pigs, and hamsters. Now, mice, Swiss albino mice, bulb C mice, C57BL6 mice, that is the black one, B6D2F1 mouse. And rats, I have Wistar, Sprague Dolly, that is 2 strains of rats, New Zealand white rabbits, guinea pigs are Duncan Hartley strain, and Syrian hamster. So these are the available that are available with me. If any different kind of animal is required, we could acquire it for that particular experiment and conduct the studies. So acquiring it from registered breeders is as per regulations, that is CCACA regulations. So unless the breeder is has got a license, I cannot purchase from them unregistered. So everything is, we have to follow the law, we have to follow the regulations. That is how we can, that means nobody is going to later on point out that your work that you did or the research that you conducted was not, I've forgotten the word. Can somebody help me? Was not illegal. Simple that I remembered now.
As per the guidelines.
As per the guidelines. So nothing, I didn't miss out anything as per the guidelines. So that means no student will have a problem that the study was conducted here and we didn't have this regulation, we didn't have that certification, so the study will be invalid. That will never be the case. So now, what are the courses that we are planning? We can have animal handling courses, animal cell culture courses. That means in vivo and in vitro, basic 2 sections. Now, in animal handling, the route of administrations, different routes that we require, whatever the drug or the test item or test material, I will say, it could be anything. So that is what would be the end use in human beings, that route we have to use in animals. So if it could be oral, it could be intravenous, intraperitoneal, subplantar, intradermal, subcutaneous, intragastric, intracardiac, intranasal, intramuscular. So whatever the intended route in human beings, that route has to be tested in animals. Because now if I have to give something orally and I do an intravenous experiment, ultimately, is it going to be used by the intravenous route? No. So the signs and symptoms or the toxicity that I see or the efficacy that I see may not be what I expect to get it oral route. So we have to choose that route. So in some cases, we can have 2 routes of administration. If I can have two formulations where I can use different routes, that also can be initially seen, which is giving the best efficacy. Then we can switch over to that route of administration for further studies, further experiments. Now that became the routes of administration. Then withdrawal of the blood sample for analysis. So how do we withdraw blood? Cardiac puncture, intravenous by tail vein in rat and mice. If it is very small quantity, the retroorbital plexus is there in rat and mice. In rabbits, we do it by the ear vein. Sometimes we can even withdraw through the penile vein. So it depends on the quantity of blood that is required and how often do we have to withdraw the blood from the animal. So there are also regulations that I cannot withdraw blood for more than 4 times in an animal. So I cannot have bleed at every hour for 6 hours. That is not permitted by CCACA. So we have to follow the guidelines. And in that case, we can have one group of animals at the odd hours, one group of animals in the even hours. So in that way, we can have the total number of say 8 hours or 8 or 10 hours that you want blood samples to be withdrawn at that particular time. So minimum, like we all know, number of animals that are required are to be used in such a way that you can conduct the statistical analysis. So minimum 6 per group in research, could be 10, could be 12, but minimum 6 is always required. If there are 2 sexes to be used, and if you know that there is a sex differentiation, 6 males, 6 females could be used. So depending on the product that we are testing, depending on the data that is available of similar product, not exactly the same, but something having the same action or something which is used in, has been used previously, could be taken as an example from the public domain and then tried out our research. See now, every time when students come here, they say, has some work study been already done? So every time it's not necessary that the same study has been done somewhere. We can have an example that, okay, a similar study has been done on different model or a different route, but we are trying to do it through our way. So most of the teachers also would say, give the reference of whatever you are doing. Every time a reference may not be available. So if we are innovative, yes, some reference could be available, but otherwise we have to plan our own. So that planning of the work or the experiment is also very important, because I have to go to the end use of the product. End use in human beings is very important, because human being is the most selfish animal on this world. What I want is, I have to be safe. So for my safety, I'm going to do all these tests. For my efficacy in any disease, I'm going to do all these tests. But if it is safe in human beings, it could be safe for my larger animals. That is my pets, dogs, cats, cattle, buffalo, horses. So these are also the animals which require to take all these medicines or all these products. So that way there is also a ratio of giving the dose or the conversion dose from rat to human or mouse to human or whatever it is. So that is also there. So if I do study on animals, it becomes easier for me for the conversion dose and use it in human beings. Then we have dissection and isolation of organs. Because now suppose there is a person who wants to say work on liver hepatocytes. So once I open up the animal, if I don't know where the position of the liver is, I may take some other organ, start getting the cells of that organ, and ultimately it will be that it is not of the liver, but it is of the kidney or so. So dissection, where you know the location of the different organs and how to isolate those organs to further either for disease inducing and analysis in the in vitro cell culture. Now this is just some pictures. All the time seeing only the letters makes us, difficult. So the picture, this is how oral dosing has been done. This is the subcutaneous injection of the mouse. This is the intravenous injection in mouse vein. And this is rabbit. We are removing the fur of the rabbit, and we can apply on the skin. We can give the injection through the ear vein. We can give the injection subcutaneous. So all different kinds of routes which can be seen. The rabbit eye is most of the time used for ocular reactions. Intranasal can be used for rat and rabbit. Depends on the volume of the drug that you want to instill into the nose. So these are the different routes of exposure. As I said again, that is always to be decided on the end use of the product in human beings and the duration of use in human beings. Next.
Please.
So this is just a screen for giving you the different kinds of instruments that are present in the institute. All of you are actually invited to come and visit the laboratory. These are not just pictures; they are available with me. So I will give you the lab address. So anybody who's interested can come Monday through Saturday, 9:30 to 6 anytime, other than maybe national holidays. Or if you can inform me beforehand, we will wait for you. Now this is a non-invasive blood pressure apparatus where we can see the blood pressure and heart rate of the animals before and after, because see the sensitivity would be slightly less compared to your carotid artery. When we do a study where we are inserting the cannula in the carotid artery and getting the data, there I may have that data and this data may have a slight difference, but I can use this animal, which is because of non-invasive blood pressure apparatus, the reaction in the same animal. Before, if I've induced hypertension, what was the hypertension? And after giving the drug, what was the blood pressure? So that becomes before and after. So that is much more efficacy, showing efficacy, other than using two different animals at different time points. And the plethysmometer is for measuring the inflammation in the rat for edema. This is a rotarod test. These are some kinds of instruments which are, I think if you are from the pharmacy colleges, you all have all these instruments in your college. Next. And this is again different animal studies that we conduct. depending on the requirement. Right from there are about, I have given a big list, but then this list will remain with you when the presentation will be with you. So you can find out what experiment you want to conduct, or if there's another something new that you want to conduct, that also disease we can try and induce in animals, maybe in rabbit, maybe in rat, depending on the blood that are required to be withdrawn at different time points and the sensitivity of the animal to that particular disease. Now, like I said, in mouse, there is absence of gallbladder. So if I have to do something on, mouse has got the presence of gallbladder. In rat, the gallbladder is absent. So even though I require to be withdrawing blood at different time points, I require a larger animal. But if I have to study on gallbladder, it has to be in the mouse. So I require more number of animals. So that is how we plan the work. So the first thing is, what is the experiment to be conducted? What is the research to be done? Which animal model to be selected, which is most sensitive, which is more relevant to the end use. So that is how the planning has to be done. Number of animals that I use, the duration of treatment, different time points analysis. So that all is also very important. Next. So now the areas of research, like I've mentioned here, pharmacology, yes, molecular biology, microbiology, and in vitro research activities. Now in microbiology, actually we can do some of these tests, but my lab is not approved by any microbiological, say, what would you call it? Recognition as from any microbiological department or so. These are things which are normally done, if they can be done. So if it is to be as submitted to any regulatory authority, it cannot be done, because my research institute cannot submit any data to regulatory authorities. So whenever we are doing preliminary work, it has to be done through APT Research Foundation, because there we have to go through initial studies and then decide on the final studies, and those kind of studies then can be done at APT Testing and Research Private Limited, because APT Testing and Research Private Limited is FDA approved. APT Testing and Research Private Limited is FDA approved. It has got ISO certification. It has got my CCSEA. We have IBSC. We have IAC as per CCSEA, and those regulatory certifications are there for the company. In my APT Research Foundation, we have CCSEA, IBSC. That is the requirement, basic requirement. That is all that we have. So these are the different kinds of experiments that we can do in animal models, which have already been done many times in my lab. This is nothing new. It has been done since 2006 till today. We have been doing different kinds of studies. So this has been done. Another important thing that I'm looking into now, there could be so many people who are listening to this, the imaging systems, imaging of the different kinds of diseases that I'm inducing. Just now I'm giving you the photographs of the animal, whatever has been induced after opening up the animal, before, after. But I'm looking for imaging systems where it could be an MRI, it could be an X-ray, it could be a fluorescence technique, where the photographs come into picture, where nobody can defy that this has been done and it was before, and what were the results after the duration of the treatment. Why I want this? Because I'm working mainly on natural products, and if people don't believe in natural products, this is the only way I can convince them that this was the photograph before and this was the photograph after. Now, when we do a study, usually what are the groups that we use? We use a control group where they are normal animals. There's a disease control group where the disease has been induced in the animals to show that yes, the disease was induced in all the animals under study. We have a group where we use the standard drug of treatment that is already there in the market. And we have maybe 2 or 3 groups of the test drug or test material, the test compound that has to be tested at a low, intermediate, and high dose, or already some study has been done. It could be taken, 1 or 2 doses can be selected to see the efficacy of the product first. So once the efficacy is there, then we plan how to do the toxicological study. So these are the minimum number of groups that are required. Every student who comes, the first question is, what is the cost of this study? I said, see, it depends on what you want to analyze when you're doing the study. Not only the number of animals, the duration of treatment, and analysis at different time points. If I'm going to analyze only twice, I'm going to charge you for twice. If I'm going to analyze 4 times, charges will be for 4 times. What I would like to bring to your notice is whatever charges I'm charging to the student is the actual cost. It's not like the cost is 10 rupees and I'm charging 100 rupees. It's never the case. Maybe I may charge 11 rupees because I have to run my institute, but I'm not charging 10 rupees cost and I'm charging 100 rupees or 200 rupees. No, it never happen. You all can check anywhere. And another important thing is whenever the study is going on, the student or the faculty can always come and see how the work is being done. There are students who come and remain with me for the whole study. Sometimes they come at different time points. Okay, this is the way I dose the animals. There's an observation of 4 or 5 days. You come after 6th day. Again, we have to take the body weight, food consumption, whatever parameters we are studying, come when we are doing that. So that means you know actually what we are doing. And at the end of the study, when I'm sacrificing the animal, the student has to be present so the student can see what actually is done. Because see, every time the student is not trained to do all these things. My staff is trained to do all these techniques by these techniques. So there's very little cruelty that CCACA says. Unless the person is trained, you cannot handle animals. Unless the person is a graduate, you cannot handle animals. So my staff, we already have trained them. We have the certification of their training. So they are doing these studies on your behalf. But you have to be present there to see. If you want to, if you're a PG, yes, I do train you how to dose the animals by the different routes. You can open up dissection, do the dissection of the animals. You can do the weighing of the organs. You can do analysis of blood. All that can always be done by the student. So in fact, I want the students to come and do this themselves, because when they are facing their viva or their examination, they have to be confident that what they have done is right. I am confident, but what about the student? Because if the student is sitting in the dark, not seen anything, he or she cannot answer, and then there is a clash that whether you have really do you know what the research you have done. So that also can be prevented. So many a times students do come here and see how their experiment is, or either if they are permitted to do the study, if they are a PG, they can handle the animals and they can help us during the whole experiment. Now, in vitro research, we can conduct, I have written here, monoclonal, polyclonal antibodies, cytotoxicity by MTT assay, adipocyte differentiation assay, mast cell degranulation assay, and nitric oxide, pancreatic cell isolation, glucose uptake, and scratch assay for wound healing. So these are all the different in vitro studies that we conduct, where you can see the efficacy in different diseases. Now, raising of polyclonal antibodies is a standard procedure where we take about 45 days for getting the first batch of anti-sera. Monoclonal antibodies raising takes a longer duration because it is very specific and it could have slightly changes in the activity that we are doing. Now, cytotoxicity is after we see the efficacy. Now, as in regulatory toxicology, we require in vivo and cytotoxicity. So that is included here, so that the chemical that the person is going to, researcher is going to use later should at least have preliminary safety in the in vitro, where we use L929, that is the mouse fibroblast, for conducting the study. That is the as per regulatory toxicology. And then we do the MTT assay and the different kinds of assays. Now, adipocyte differentiation assay is done for anti-obesity drugs. Mast degranulation assays is done for asthma or inflammation kind of diseases. Pancreatic cell isolation and glucose uptake sure for diabetes. Scratch assay is done for wound healing. That is there. It is known. So we can do a scratch assay for wound healing and then do the in vivo assay in animals, in rats, to see that we are getting similar results. Now, if I have an array of chemicals to be tested, initially I can do the test on in vitro and then select the ones which are showing the best efficacy and go for in vivo testing. When I come to molecular biology, actually, I'm not a molecular biologist. When I studied, I think very little of molecular biology was there. So I'm not zero, but at the low level of molecular biology. I have experts who know that. So I wouldn't consider myself to say something very much more in molecular biology. I'm extremely sorry for that. Next. Now, when we do research on plants and in herbals, plants and herbals, why I'm saying herbals, because some of the plants may have some references in Ayurveda, Unani, homeopathy, and some may not be having any references. So this is how the research is started at Research Foundation. Plant authentication. That means I would give an example that, okay, I want to study on a particular plant. I've brought a plant, a leaf or a stem, and I suppose it is, say, Tulsi. I will take an example, Tulsi. I start working on it, I do all the work and I say, okay, Tulsi has got this property, Tulsi has got that property. And when I go for authentication, I find that it is not Tulsi, but it is Sabja or it is something else other than Tulsi. So that should not happen, because when I'm going through the review of literature, everything is I'm looking for Tulsi, but actually the plant that I have in my hand is Sabja. So there is a difference in it. So plant authentication is the first step that has to be done. So that means identification is very important. Then I can do my review of literature. Regarding that plant which I have selected, whether what kind of studies have been conducted, what kind of research needs to be done, and depending on their, now in what happens usually, chemistry departments do analyze a lot of the molecules that are present, the structures of the actives that are present in the plant, maybe leaf, stem, root, flowers, but they never go up to the biological activity. And if I can see that this plant contains, so these are the molecules that the plant contains, or the principles that are present in the plant, based on that, I can predict or try to find out that, okay, it could have this activity against this disease. So that is how chemistry, when I'm looking by the review of literature, I can consider that and plan my experiment on that plant. Now, need for preclinical efficacy and safety is, I've already said, that is very important. That has to be done after getting the review. Selection of the appropriate animal models, because like I said, if I don't select an animal model which is sensitive to that particular disease, I will be failing in doing that animal experiment, and the time and money all will be wasted, whoever it is, maybe student or in-house. So now there are, suppose I have Swiss albino mice and bulb C. Now bulb C is supposed to be having more prone to induction of cancer. So I should use bulb C for that particular disease and not use Swiss albino mice. They could be, now we have in knock-in, knockout strains which are also available, but those are so expensive that it is very difficult for a student to conduct research there unless he's sponsored by somebody. So that sponsorship, if it is available, we can go right to the particular genetically modified animal and use that for that particular disease. Now we have to have the assurance of the quality of the product or the plant extract or whatever I have done. So there are different tests to do that quality of the product. I have to avoid the repetitive research. When I go through the review of literature, I see what is the observation. I can or I may conduct a preliminary pilot study to see whether I'm getting what has been mentioned somewhere in some other paper, because nowadays that also becomes a question mark. Many times the repetition by some researcher may not be available, or I may not get the same readings. There could be so much of variation in that. So it's better I have a pilot project and then go for the final project. And what I do is I always compare with a marketed sample or marketed preparation for maybe modern medicine that is using for the disease, or whatever is already used by the herbal preparation, Ayurvedic, Unani, homeopathy, different kinds of the different herbals that are used in as medicine. So these are different institutes where we have already done work for so many years with all these people, all the different companies or institutes, research institutes, where I'm highly obliged because they have helped me in this. Collaborations we have with different colleges, they also require, so that the students can come see whether I have a collaboration or not. Every student who comes to me will have the same treatment, but if he gives the name of a college and if he's a student of that college, Yes, never going to say no to anybody. But some of the colleges we require collaboration, so those agreements have been signed. This I have come to the in vitro studies. I've already mentioned these are the in vitro cytotoxicity tests that we do, the different cell-based assays, sulfate reducing bacteria assays, cell adhesion assays, cell fusion assays. Now, these are toxicity studies and the biocompatibility and pharmacological testing. So, all these, like we do in vivo, the similar studies have been done in vitro. As per requirement of the student, the studies have been planned and conducted. Now, in normal cell line, now when we do any now these are the different cancer cell lines that we use for. So, when we conduct these studies in vitro, we always compare with a standard drug or a positive control, showing what is the percent kill of that particular cell line compared with the new product that is to be tested, new test material that will be tested. Now, after doing this cell, now as the cancer cells are being killed, we have to find out whether the killing effect is because of the toxicity or the efficacy to kill the cancer cells. So then we do the study in L929, which is the normal cell lines. So mouse fibroblasts. So if I find that the kill in normal cell line is less, then I can say that yes, the activity effect is on the cancer cells. Well, many times what happens is I got 80% kill here, I got 90% kill in the cancer cell lines. But when I do a test on the cell line L929, there also if I'm getting 80 and 90%, it is the toxicity that is causing the death of the cells and not the efficacy. That particular has to be noted by all the students. And the cell culture and the types of culture that we use we can get it from plants or animals. Usually we have done it from animals. The different cell cultures, primary cell culture and the subculture. So these kind of things, I'm not trained in it, but my staff is trained in this because my techniques I'm vivo person. So it will be difficult for me to conduct such experiments, but my staff is there who have done biotechnology and who know all this. And the morphology of the different cells that we are doing here, that all of you must be knowing. The fibroblast, that is that's the first picture. Second one is the epithelial cells and the lymphoblasts that are seen. So by looking at the cell shape, size, you can always say whether they are normal or abnormal, whether it is cause toxicity or it is not cause toxicity, whether it is shown kill or it is not showing kill. So now this is the fibroblast cell line, mouse fibroblast, here L929, where it is a normal structure. And after treatment, if it shows like this, it is going to show that it is showing the toxicity. Before treatment of MCF cell line, this was the breast cancer cell line. After treatment, what is happening? So when this same drug or the test material shows safety in L929, I can go ahead blindly to be used in for further testing. These are the students who have conducted their PhD, finished their PhD through APT Research Foundation. Their names, and actually I have not given the topic of, yes, the topic is there. So there is nothing, go to the next. There is, no, so there is, always the selection of topic or the area of interest has been left to the student. I don't say, okay, I want to work on the brain. So I don't tell all my students to work on the brain. The student will say, I want to work as a hepatoprotective, something to do with the liver. Somebody says, I want to work with the hair. Somebody says, I want to work with anti-obesity and obesity or diabetes or whatever. So that's why if you see, the topics of research are totally different in all the students. Because if I give you a topic which is of not interest to you, your work interest is so little, I am not satisfied with the research at all. Just getting the degree has not that much of a meaning from my point of view, but you should also have keen interest in doing the research that you are doing, so that research that you have done is going to be used later for human beings, as a social cause. So like if you see anti-obesity, anti-diabetic, anti-obesity was a plant, anti-diabetic was a fern, Antihistaminic also was a fern. Wound healing was a combination of 3 plants together. Hair growth inhibitor was a plant. Hepatoprotective was a plant, single plant, and also hepatoprotective was for microbes. That is your lactobacilli and all that. So that was also hepatoprotective. Anti-cancer was again plants. Osteoporotic, anti-osteoporotic was also of a plant. Hair growth promoting activity were of 2 plants. So as I'm not a chemist, I cannot synthesize any new molecules. Working at APT Research Foundation is with natural products. So that's why I say natural products would be the root, stem, leaves, fruit, seeds, or the whole plant, availability, efficacy, and safety. So that was of importance to all the students who have done their research. This is patent granted and funded project. We have done one BIRAC project, Big Four, and SERB project. We are doing one that is going on. The patents filed by Mrunmay, who has done a PhD. This was the composition of weight loss, which is now over. Next. And these patent applications were submitted, but we didn't get the final patents done. Next. The publications are there, like ma'am said, 30 publications. So I'm not aware of the number of publications that I have. But whatever we use, we try to publish in the research papers so that whatever we have done goes ahead. Some books have been written a long time ago. The future. Now, what is the future of all the whatever research I have already done, the students who have done, my students, or I was interested in doing work. So those research, when we got good efficacy and safety, we have converted actually those products into tablets or capsules with permission from FDA, Maharashtra State FDA, and we have got a license for manufacturing. And I also manufactured quite a few of them, which I actually am not selling at this moment, but it as a social cause, it is given to people who are suffering from that particular disease. And as we have already done the safety, it has been given through doctors who are in that field. So they also monitor the patient before and after. And that is what is my main aim. Producing theses and putting them in the cupboard has no value. But even if out of the 10, if 5 are converted into medicine to be used by particular route that we have studied for the good of our people, I would say, I come from Pune, Maharashtra, India. So that is my aim. So some of these products could be exported to other countries because they don't have these kind of plants that are available in India. There could be some cousins of those plants, but then that will be a next stage. But if we can do research in such a way that our main aim is to manufacture a product to help our people, our own people, and these products, actually, if you see, the cost is very affordable compared to other drugs and having less toxicity. I would not say no toxicity at all, right? Because if the people, if I'm telling somebody that you take 1 tablet in the morning and 1 tablet in the evening, and when the patient feels better, the patient starts taking 2 in the morning, 2 in the afternoon, 2 in the evening, there could be a toxicity, but it is not, it is mentioned on the label, take 1 in the morning or 1 in the evening. So that control is not with me. So that's why we have to see to it that, or I say that it could have some toxicity. So as it is also in allopathy, we also already mentioned there, if there is any adverse effect, go to the doctor. So the same thing has to be followed for herbals also. And the main aim is to convert our research into IPR and then make it marketable, make them into marketable products to be used as medicines, nutraceuticals, or health drinks. Making the thesis and keeping it in the cupboard and not utilizing it for anything. Because see, we have ultimately wasted, consumed our time, money, efforts, and of course, discussions and whatnot. So that should reach the market as quick as possible or as suitable to the particular disease sector. Now, the data for products would be in the form of efficacy and safety in animals. And if it is safety in animals, we also have conducted some case studies where if I have a dermatologist as a friend, I will tell him or her, okay, this is my product. Could you please try it on your patient for wound healing? So they do try it out and they get the results and they refer it back to us, give us the data before and after. So if that a few case studies are done along with the thesis, it shows that yes, it is showing similar activity in human beings also. Because there's always a question that whatever studies we do on animals, are they really relevant or exactly reproducible in human beings? Yes, they are not exactly reproducible, but some minor modifications could be there. But otherwise, the data that you get, it will never happen that I've got a wound healing cream which is effective in animals and it's not going to have any effect in human beings. That will not happen. Or I've got a hepatoprotective drug which I've done different models. So many animal models I've done to induce liver disease, and when I give it to human beings, it is not showing any efficacy. That will never happen. And final aim is to use our material in India at very affordable prices and export to other countries. Most of the times what happens is we export products, raw material to other countries at X cost, and they sell it back to us at maybe 10x or 20x or 50x or 100x. So instead of that, if we can manufacture our own at affordable cost, that is going to help our country. So that is of most importance. And when we export to other countries, we can have, suppose I'm charging say 400 rupees or 500 rupees for a bottle, I can very well charge them in dollars or euros, where the cost is going to be more, because for them maybe $100 or $50 is not much of an amount, where for us it is a very high amount. So this kind of thing, so we can improve our what would you call it, use of our products through other countries. Naturally, India will be at the top. I think I've come to the end of my presentation. Hopefully it was well in time. I'm not sure. So I thank you all on behalf of APT Research Foundation. This is the contact number. That is the mobile number of my lab. And there are 2 email IDs. So anybody requiring any information, you all can always contact or send an email with your contact number. So we can call back or send an email later. I think that is all from my side. So please, all those who are interested in doing research in different kinds of diseases, do come to me. Whatever best we can do, best model that I can develop, I will do it if it is not developed before, and you can go ahead. So I thank you all for giving me this opportunity and listening to me. Patient listening. It may have been a repetition for some, but for me, I would thank all of you to give me this opportunity again. Thank you very much. And open for question and answers. Thank you very much.
Thank you, ma'am. Thank you for such an informative session. Ma'am, we have a question from Prakash Almani, sir.
Yes,
His
sir.
question is, what are the model systems to be used for probiotics preclinical study that are very close to human beings? How much it costs for commercial purpose?
Cost I cannot say. You want the rodent model to be used for probiotics. Now, when we use initially for probiotics, there are the 3 steps where you have to go for bile tolerance, acid tolerance, adhesion. Once they are following these, if they are passing these parameters, then you go in for the animal studies. So you can give them dose them to the animals, see the different, by doing the different assays, you can see what is the effect. Now, cost for commercial purpose would be after finishing the study, what all cost expenses we have done, that would go for the commercial. I'm not too much into commercial, but I can give you, if somebody wants this, you can give me the number of strains that are to be used. I will give you the number of doses and the groups that are used, and based on that, I will give you the cost for conducting that particular study. See, every research is not the same, so the cost is going to differ. Somebody's research could cost 20,000, somebody's could cost 50,000, somebody's could cost 80,000, maybe 1.5 lakh or whatever it is. So depending on the experiment that has been planned, depending on the analysis that is to be done, the cost will come for each and every study.
Any more questions from the participants? You can please unmute yourself or text it in the chat box.
Megha,
Yes, sir. Good afternoon.
I want to make a comment.
Yes, sir.
First of all, I'm very thankful to APT ma'am. And personally, I visited
Yes.
her APT Research Foundation.
Yes, sir.
I thanks first Dr. Bodhankar because he made me
He was
to
the link,
come at
right?
your institute.
Right.
And really, you are doing a wonderful job in the microbiology and testing and helping to the many students in pharmacy college. Now our microbiology society, the reason I have told Megha ma'am to arrange your lecture is the same, that we want to utilize
The facility.
the various testing in your laboratory.
Yes, sir.
So today's lecture is recorded. It will be circulated throughout the India. And definitely today is the beginning.
Yes, sir.
Slowly and slowly, microbiology society will come more closer to your organization. And together we'll do many things.
Yes, sir.
Thank you. Thank you, ma'am.
Thank you
for
very
coming
much, sir.
on our platform.
Thank you very much, sir, for your kind words. And like I said, when we do even studies on animals, like we did a study on wound healing. So whatever we applied, after that, I told the students, you take the swabs, see something related to microbiology. So what were the organism that you found? What was the number that you found? That will give you the antimicrobial activity as in vitro. And you can also see the wound contracting. So they go hand in hand. So this is how we are going to correlate microbiology with efficacy and safety.
Very correct. Very correct. So I really appreciate Megha
Yes,
for
thank
arranging
you very
this
much.
lecture. Actually, today I am in Cambodia.
Oh, wow. Great.
Yeah, Phnom Penh. But even then, I was very eager to attend your lecture.
Thank you very much, sir. Thank you.
Excuse me, Dr. Kishori.
Yes, sir.
Can you please focus on the training programs, because the students are there. That is necessary.
Kishori ma'am, you have muted yourself. Yeah.
So the different training programs are there where we can have maybe a schedule for 7 days, 15 days, 1 month as required by the students. So if I have a batch of maybe 10 students, what is their requirement? We can have arrange different programs. See, everybody may not be interested in doing a work for 1 month. Somebody may have 15 days. So in vivo, in vitro, both can be done for the students at different time points. And also the time that is the students have for their in through college, when they have their vacation or after finishing their graduation, there is a time for them to still finish, get admission to post graduation. That time they can do all these kind of training programs, which is going to help them in their future. Also, there are students who come here and who want to go further to different countries to do their studies. So here the basic training is given, hands-on training to the students, which will help them do better when they go outside India. So that was the main training program that we were planning, depending on the requirement of the student and the time the student can give us. See, because if I have to train an animal for in vivo handling, a student who has never seen a rat or a mouse will take 8 days just to go close to the rat or the mouse. Then training in handling and giving the doses and then withdrawing the blood or different body fluids. So that cannot happen in 10 days. I will have to have that for 1 month. So depending on the end use of the requirement for the students, the training can be scheduled anytime that is required. The in vitro can be done by all undergraduate students also. So actually our aim is to give hands-on training to all the students. The theory is taught to you in college, you can read, but the practical work that you do is of most importance. And so that is our main aim, to give you practical training for the in vivo or in vitro that you require. That would be my contribution to all the students, depending on the duration that they have. Now, sometimes the student may have done some animal handling, but he doesn't know how to withdraw the blood or collect the urine or get the CSF or something like that. So that can be done in a shorter period of time. So if that person's batch or a student group is about maybe 10 students, I can give them a training program for 1 week. So the cost will depend on what I'm teaching and what is the duration of the training. That way, we can put it up on the site or give it to people here, sir, in our society. So people will know exactly what the cost would be. And like you had just now this online exposure, that way also can be taught, maybe shown by pictures, but that would be initial, basic. Ultimately, by just looking at the pictures, we don't become experts. So doing hands-on training would be the best thing to do.
Thank you, ma'am.
Yes, sir.
I guess there are no more questions. I would like to thank Dr. Kishori Apte, ma'am. The very first line you said, ma'am, the APT institution is for the people, for the students, by the students and from the students. The sentence itself shows your dedication and your sweet gesture towards the research scholars and students and to help them out in their difficult task. So
Yes.
it's really a pleasure to have you for today's session, and we'll make sure that we'll approach you at the earliest, and the students, the research scholars, as well as the students will get more and more information from your side. So I'm really thankful from MBSI entire team for spending your time and giving us such a informative session. Thank you, ma'am.
There's no waste of time. Actually, it is not spending my time means it is very useful, because
Yes, ma'am.
these things are there in my brain. I don't have to prepare anything for this, because I'm in this field for the past now 40 years. So if
Yes,
you
ma'am.
wake me up from my sleep and ask me some questions about toxicology and efficacy, I think I should be able to answer, other than in vitro. That's what I've been saying, because I never learned in vitro. So I'm not an
Yeah.
expert in that, but I have experts who are with me. And microbiology, yes, we did in MSc pharmacology also, I did learn microbiology. So that is also of close to me. It's not that microbiology is not, I don't like microbiology. I like microbiology also, because now most of the diseases that we suffer from are because of the microbes in our gut. That is such an important thing. So microbiology
Yes,
has got such an important, requirement, that people still don't know what is the use of microbes in our body.
truly said,
That's
ma'am.
where we
Truly
have to
said.
go ahead. Right.
Yes,
Thank
ma'am.
you very much, Meghna ma'am, for all this information and the time given to me. And like we can have these kind of sessions more often also with different
Sure,
students.
sure, ma'am.
That could be fine.
It's our pleasure, ma'am. I would also like to thank Renu Mukherjee, ma'am, who has been a very good coordinating person in between Dr. Kishori Apti ma'am and myself. So I would also like to thank Renu Mukherjee, ma'am. I would also like to thank Dr. Deshmukh, sir, who always keeps on pushing me that we shall conduct few sessions for research scholars and let us get more and more knowledge about the subject. So I would also like to thank Dr. Deshmukh, sir, and last but not the least, all the participants who have joined for the session
Yes.
today.
Yes.
So thank you, one and all.
Yes. And from my side, thank you, Dr. Deshmukh and Dr. Bodhankar, who was the link between us. So he has brought us all together. So a big thank to all of you. Thank you to all of you.
Thank you, ma'am.
Bye, sir. Bye.